So good evening everyone. In today's video lecture we will discuss Telangana EAPCET 2026 Botany paper analysis. So this exam was conducted on May 5th, shift 1 paper. Okay. Yeah. Yes, Botany paper. That first. Okay. So here read these questions. So unit of marker used in creation of three domains. Here, so which marker we use to create three domains? By whom we will discuss. So here explanation. Carl Woese. He used 16S ribosomal RNA sequence. He used 16S ribosomal RNA sequence as a biomarker for evolutionary studies. Mainly for phylogenetic means for evolutionary studies. Why he used the 16S RNA sequence means as a phylogenetic? Because of you need to classify living organism into three domains. How many domains? Three domains. So there first one here three domain. Like first domain archaea domain, second one is bacteria domain, and the third one is eukarya domain. So likewise there are three domains were proposed by the Carl Woese by using 16S ribosomal RNA sequence as a phylogenetic marker. So here in which archaea domain include archaebacteria group only. It is a primitive bacteria. It include archaebacteria group only, and bacteria domain which include a kingdom called Monera kingdom. Only Monera kingdom which include in this domain. Monera kingdom. Now eukarya domain include four kingdoms. Very important. Eukarya domain include four kingdoms. So they are first one Protista, and second one is fungi, and the third one is plantae, and the fourth one is Animalia kingdom. So likewise Carl Woese proposed three domain by using the 16S ribosomal RNA sequence. So here right answer is 16S ribosomal RNA. So next one here choose the incorrect pair. Here asking incorrect pair. We have to choose incorrect pair. So here giving elements and their role. So here zinc oxygen synthesis, nickel activator of urease, molybdenum component of nitrogenous, manganese activator of catalyst. Okay, let's discuss. So which statement or which pair and elements their role is incorrect. First we will discuss about zinc. Here I am writing about zinc. Here zinc is a very important mineral elements, so which is helps to synthesis of oxygen synthesis. So A is correct pair. And a little more about gin is also act as a activator of various enzymes. So like they are also activator of carbonic N enzyme, and they are also activator of alcohol dehydrogenase enzyme. Simply we can use ADH, and also carboxy. So these are the few hormones which is activated by this zinc. Remember. And do you know the deficiency of zinc? Deficiency. So deficiency of zinc in plant which leads to diseases like little leaf disease and also Kyra disease of rice. So these are the two deficiency disease by zinc. Okay, fine. Now coming to nickel. Here I am writing about nickel. So nickel is the most important, and it is the last discovered elements among 17. Generally there are 17 elements are essential for plant growth and development. Among 17, the last and 17th discovered element is only nickel. So here about nickel, generally this nickel which is activator of urease enzyme. Important. Urease enzyme. It is activator of urease enzyme. Do you know this urease enzyme was discovered by James Buck Sumner? Here remember, this was discovered by James Sumner. Remember, James Sumner discovered this urease enzyme from Jack Bean, and it is a activator of urease enzyme. So here we need to know that nickel is activator of urease. So this statement is also right. Now coming to molybdenum, here I am writing about molybdenum. Yeah, molybdenum. So about molybdenum, it is a component of nitrogenase enzyme. It is a component of nitrogenase enzyme. So this nitrogenase enzyme play important role in during nitrogen fixation process. Okay, so not only it is a, along with molybdenum, iron is also. So molybdenum, iron, both are the components in nitrogenase enzyme. So this enzyme play important role in nitrogen fixation process, biological nitrogen fixation process. Okay, remember molybdenum. And deficiency of molybdenum which leads to some kind of disease. Here I am writing disease of molybdenum. Whiptail of cauliflower disease. Here I am writing whiptail of cauliflower disease. and also one more disease caused by the due to deficiency of molybdenum, that is dieback of shoot. So these two disease which is caused by the molybdenum. Here remember generally catalase enzyme. So one more important enzyme here we need to remember, catalase enzyme. So this catalase enzyme require iron. So iron ferrous or iron is essential for the activator of catalase, very important. Okay, and here coming to manganese, here giving the manganese. So manganese is activated by catalase. No, this statement is wrong. Why? Because the activator of catalase who? Iron ferrous. So here the wrong answer is D only. So D is the wrong and incorrect page. Okay, fine. And here so we need to remember few important things about some more elements, they play important role. Here we will discuss few more important elements and their role. But here in these questions the right answer means incorrect pair is D, that is manganese. It is not a activator of catalase, it is activator of activator of iron is a activator of catalase only, remember that one here. So here we will discuss about manganese. Okay, right. So manganese which is involved in photolysis of water, very important, which is involved in photolysis of water means the water will undergoes breakdown. Like if water undergoes breakdown, like it produces 2 electrons plus 2 protons plus half oxygens. So generally this photolysis process which occur in lumen of thylakoid during photosynthesis process. So along with manganese, for the photolysis of water there are also required, there are 4 manganese molecules are required for photolysis, and also 2 calcium ion and also 1 chloride ion. So these are the few elements which is essential for photolysis of water, and these photolysis of water are occur in the lumen of thylakoid of photo. And not only it is involved in photolysis of water, and this manganese is also activator of few enzymes. Also it is a activator of few enzymes, like it is a activator of very important enzyme, that is exo enzyme. So exokinase enzyme which activated by this manganese, but not manganese activate this catalase, but who catalase activate by iron, ferrous or iron. Okay, so remember all this. And due to deficiency of this manganese, so due to deficiency of manganese which leads to a disease called gray spot disease of wood, is due to deficiency of this manganese, remember. And also we need to remember one more important mineral elements, and it also play important role in plant growth and development, that is magnesium. Here I am writing magnesium, about magnesium, Mg2. So generally magnesium is an activator of various enzymes, like it is an activator of pepcase enzyme. Pepcase, generally this pepcase enzyme phosphoenol pyruvate enzyme which is involved in carbon dioxide fixation, like which fix the carbon dioxide with the 4 carbon compound, that is pep, sorry, 3 carbon compound, that is pep, 3 carbon compound pep. So then it result to form 4 carbon compound, that is oxaloacetic acid. 4 carbon compound will form by involving pepcase enzyme, and it leads to C4 cycle. Okay, so this pepcase is activated by this magnesium. Okay, fine. And one more, not only pepcase but also it is the activator of rubisco enzyme, very important. Rubisco is the most abundant enzyme in the world, and this rubisco enzyme get activated by this magnesium. How means generally this rubisco also play important role during photosynthesis process, especially in C3 cycle. How means this rubisco which fix the carbon dioxide to the 5 carbon compound RUBP, then it result to formation of 2 molecule of 3 PGA. So in PGA, each PGA contain 3 carbon. So hence it is called C3 cycle. In C3 cycle the rubisco play important role. In C4 cycle pepcase play important role. So and these two enzymes are activated by these magnesiums. And not only that, and also magnesium also which activate the hexokinase enzyme, very important, hexokinase enzyme. Just now we discussed hexokinase enzyme activated by 2 important elements. Okay, so not only that magnesium, but also just now we discussed now manganese. So manganese and magnesium both activate this hexokinase enzyme, very important. So generally this hexokinase enzyme where they play important role means in respiration, especially in respiration. During respiration what happen? So here glucose get converted into glucose 6 phosphate. So there we have hexokinase enzyme. Later fructose 6 phosphate get converted into fructose 1 comma 6 bis phosphate. There also we have hexokinase enzyme role. So likewise, this hexokinase enzyme play important role during glycolysis process in respiration. So this hexokinase enzyme get activated by this magnesium. So likewise, hexokinase, rubisco, peptase. So these are the various enzymes which is get activated by this magnesium. So this is all about the various elements and their role. And we move one more important MCQ, asked MCQs here. Third one. Yeah. Okay. So here third one, the biosynthesis of ribosomal RNA occur in. So here in these questions, where ribosomal RNA synthesis occur. So here read the option. Ribosomes, Golgi bodies, glycosome, nucleolus. So let's start some explanation about these questions. Here generally we know that the brain of the or the center of the cell is the what? Nucleus. See, this is, if you think it is a cell, and the center of the cell is called nucleus. And nucleus itself has some small spherical like structures. See, this is called nucleolus. So what is the location of nucleolus? Which is present inside the nucleus. So organelles within the organelles. Already nucleolus is a membrane bound, a membrane and cell organelles like ribosomes, like ribosome, and also centriole. So centriole, ribosomes, and nucleolus. These are the three important cell organelles. They do not contain any membrane. Hence, they are called amembranous cell organelles. Okay, fine. And these nucleolus, which is present like a spherical shapes, spherical in present in nucleus only. Present in nucleus. The main role of this nucleolus is it helps to synthesis of ribosomal RNA. So very important. The nucleolus important role is to synthesis of ribosomal RNA. Hence, this nucleolus is said to be as a factory of ribosome. Okay. So here right answer is nucleolus. Yeah. Right answer is what? Nucleolus. So then we move on. Another important MCQ here. Yeah. Fourth one. Yeah. Fourth question. Yeah. Here, nodules of soybean export the fixed nitrogen in the form of. So these questions which ask mainly from nitrogen fixation. That is biological, especially in biological nitrogen fixation. There are generally these biological nitrogen fixation occur in leguminaceae plant, especially in leguminaceae plants. So leguminaceae plant include like groundnut plant, groundnut and soybean plants. Okay. So here in nodule of soybean, generally this in leguminaceae plant there are nodules are formed inside the root nodules, inside the nodule. So this formation of nodules play important role by a bacteria, a symbiotic bacteria or symbiotic bacteria. The bacteria name is rhizobium. Very important. So rhizobium bacteria play important role formation of nodules in root of leguminaceae plant for biological nitrogen fixation. Okay, fine. Right. So here asking what? So in soybean, the formed fixed nitrogen export in the form of option proteins, organic acid, urides, and amines. So here urides. Why urides are right answer means we will discuss here. Yeah. So here generally in a just wait. So in plant like soybean, the fixed nitrogen in root nodule is exported to other parts of the plant, to the other parts of the plant, generally via xylem. And not only water but mineral element also transported through this xylem in the form of urides. So why means this urate has allantoin and allantoic acids. So urides such as allantoin and allantoic acid, and this allantoin and allantoic acid has high nitrogen carbon ratio. So hence in soybean the fixed nitrogen which is exported in the form of urides such as allantoin and allantoic. So right answer is urides. So next one, fifth one, identify the correct statement. Here we need to identify correct statement related to the class Pheophyceae. Here Pheophyceae means brown algae. So first we need to read this statement. Asexual reproduction occurs usually by biflagellated zoospores. Asexual reproduction here. Here sexual reproduction is by oogamous method only. Here sexual reproduction in Pheophyceae or brown algae occur only oogamous method. Food is stored as a simple carbohydrate. In brown algae, the food is stored in simple carbohydrate. So we have to highlight these keywords in the form of mannitol and laminaria. Major pigments are chlorophyll A, C, and xanthophylls and carotenoids. Cellulose cell wall has outer gelatinous coating of algae. So in this, we have to choose the correct statement. Let's start some explanation on this brown algae or phaeophyce. Generally, in phaeophyce brown algae, here I am writing explanation. Asexual reproduction occur by biflagellated zoospore. Remember, by zoospore. Here zoospores nothing but the spore which has motile spores are also called zoospores. Means, see, this is a spore. It has two flagella. Biflagellated zoospore or motile spore only by perform asexual reproduction. So here asexual reproduction occur usually by biflagellated zoospore. So this statement is correct. Okay, fine. And now coming to sexual reproduction is by oogamous method only. Here in questions asking oogamous, but actually what happened? Sexual reproduction in here sexual reproduction occur in three method. They occur, maybe they are isogamous method, isogamous, or anisogamous, or oogamous. So they can be occur in isogamous, anisogamous, oogamous, not only oogamous method. So this statement is wrong. Only make this statement become wrong. So in phaeophyce member or brown algae, the sexual reproduction could be occur either isogamous, anisogamous, or oogamous. Okay, so this statement is wrong. Food is stored as a simple carbohydrate in the form of mannitol and lamina. No. So generally in phaeophyce member, the food stored in the form of mannitol and laminarian. But generally these mannitol and laminarians are complex carbohydrate, not simple carbohydrate. It is a complex carbohydrate. So but in question asking simple carbohydrate. Now this statement also make wrong. And here coming to D, major pigments are chlorophyll A, C, xanthophyll and carotenoid. Yes, generally here green algae contain A and B mostly, and brown algae contain A and C, red algae contain A and D. Like that we have different pigments, and along with we have other pigments like chlorophyll and not chlorophyll, xanthophyll and carotenoids. Here also xanthophyll and carotenoid, like that. Okay, so this statement is right. Cellulose cell wall has outer gelatinous called algin. Yes, so this phaeophyce member also along with cellulose cell wall, outer of the cell wall there is another gelatinous structures is called algin. So this algin also present in phaeophyce member. This statement is also wrong. In red algae generally present charagen. Okay, charagen gelatinous structure, but in phaeophyce member algin is made up of the cell wall. So this statement is also right. So here A, D, E. So AD statement is correct. So next one, here sixth question. Plasmodesmata are usually observed between. Okay, so here generally plasmodesmata. Now we discuss about plasmodesmata. So what is plasmodesmata? So plasmodesmata is a type of junction. It is a cell junction. It is a type of cell junction. Generally plasmodesmata form, it is a connection between cell, adjacent cell. Why? To transport various molecules between cells within tissue. For that reason there is a connection which form. That connection is nothing but plasmodesmata. So plasmodesmata generally seen observed in here in these options: seed tubes and bast fibers, trachea and phloem fiber, xylem parenchyma and xylem fiber, seed tubes and companion cell. So generally which is seen in seed tube cells. Here seed tube cell and companion cells. Why in this seed tubes and companion cell? We have one explanation here, we will discuss. So generally these seed tubes and companion cells both are type of cells in phloem tissue. Generally this phloem tissues is help for transport of food, and it is a permanent tissues transport food material. And phloem is generally living tissues, xylem are non-living tissues. Okay, fine. Here generally we have phloem in which 5 types of cells we have. Okay, no, we don't want to discuss all those 5 types of cells in detail. And here with in among 5 cells we have 2 important cells. One is sieve cell, sieve tube and companion cells. So generally these sieve tube cells lack nucleus. It lack nucleus, so absent of nucleus in sieve tube cells. So due to absent of nucleus in sieve tube cell, it doesn't control, it doesn't function, it does not perform its function, and it depends completely on companion cells. And sieve tubes, here for example, if you have sieve cells like this, so this is what sieve cell, and so this is what sieve tube cell, and around which we have like this companion cells. So generally companion cell has nucleated cell. Okay, fine. So here the function of sieve tube cells is completely controlled under these companion cells. So definitely there is a need to transport various molecules between sieve tube to companion cells. So for that there is a junction which is present between sieve tubes to companion cells. Okay, so your right answer is C. Sieve tubes to companion cell is the right answer. Why? Because there is must and should be transport of material. Why? Because companion cell nucleus each and control its adjacent cell, that is C tube cell, because of C tube cell doesn't contain any nucleus. Okay, next one, seventh question here. Zygote is diploid in which life cycle? Zygote is diploid. We know that zygote is always diploid in nature. So here, regardless of whether the overall life cycle is haplontic or diplontic or haplodiplantic, here I am writing regardless whether the life cycle either haplontic means n, diplontic or 2n, or haplo means n minus 2n. So regardless whether this life cycle is haplontic, diplontic or haplodiplantic, but the fusion of to formation of gamete here, to formation of zygote, there is a need to fusion of gametes. So once the gamete fusion will take place, fusion of gametes means here nothing but fertilization of gametes or syngamy of gamete. The resulted structure must and should be formed, that is zygote. Why? Because here male gamete and female gamete both having n number of chrome, n nucleus. So the resulted structure always form diploid zygote, either in haplontic, diplontic or haplodiplantic. Okay. So remember, to formation of zygote in diploid condition, there is a must and should be formation of fusion of or fertilization of gametes will happen. Okay. So likewise, so nothing to do here with either haplontic, diplontic or haplodiplantic, but always in each and every life cycle there is a formation of zygote by fusion of gametes. Okay. So here the right answer here 1 2 3 4. Why? Because here haplontic, diplontic, haplodiplantic, diplantic. Diplohaplontic means in which 2n first and n. So here 2n means sporophytic stage, sporophytic plant body, and n means gametophytic plant body. So remember, here n always represent gametophytic plant body and 2n always sporophytic plant body. So next one here, eighth question. So from the following list, choose the correct combination. We have to choose correct combination. And here, so giving substrate, list 2 giving enzymes, and list 3 product. So PGA plus ATP and this, all these questions which is framed from Calvin cycle are also called C3 cycle. So let's we discuss. Generally, this Calvin cycle was proposed by Melvin Calvin, and hence the name came as a Calvin cycle. In this first stable product which is formed C3 carbon, so that is 3 PGA. Hence this cycle is called C3 cycle. Okay, fine. So generally this Calvin cycle which occur in three major stages or steps. In that first is carboxylation. So what happened in carboxylation means general here. So here one molecule of carbon dioxide or 6 molecule from one if for example one molecule of carbon dioxide which is fixed by one molecule of RUBP, so 5 carbon compound in presence of Rubisco enzyme act as a carboxylase enzyme, then it form 2 molecule of 3 PGA. Like that, first stable carbon 3 carbon compound, that is PGA. Hence this cycle is called C3 cycle. Okay. So then after carboxylation, then it enter into second step, that is reduction steps. Generally reduction is a 2 steps process. One is first, in which phosphorylation process will occur, phosphorylation reaction will occur, then reduction reaction will occur. So what happened in phosphorylation and reduction reaction? So in here in phosphorylation reaction, yes. So in phosphorylation reaction, what happened means here formation of 2 is there na, 2 molecule of 3 PGA which combine with 2 molecule of ATP in phosphorylation in presence of this phosphoglycase enzyme. Here simply we can use kinase enzyme, and then it result to formation of 2 molecule of bis acid. Okay, fine, in phosphorylation. So first option is C, PGA. Here this PGA is combined with ATP, then it form C in presence of this phosphoglycerokinase enzyme, then it result to form bis phosphoglyceric acid. Like that, form. Okay, fine, right. So next, what happened in reduction process? Here I'm writing. So in reduction process, here I'm writing. So formed 2 molecule of bis phosphoglyceric acid in reduction, which is combined with 2 molecule of NADPH. Then in presence of glycerolate 3 dehydrogenase enzyme, it result to formation of 2 molecule of glyceraldehyde 3 phosphate by reduction process. Okay, so this second option is also right. So here bis phosphoglyceric acid means this one. The bis phosphoglyceric acid is a product of phosphorylation, and this enter into reduction process. In reduction process, there is a combined with NADPH, and then it result to formation of 2 molecule of glyceraldehyde 3 phosphate. Like that, here glyceraldehyde 3 phosphate also formed, and this statement is right. Okay, and remember here generally, and these two steps will come from regeneration step. Regeneration of RUBP, ribulose 1,5 bisphosphate. In this question will came. So here generally in this complete view, here xylulose is a epimers to ribulose. Okay, here next one is xylulose or epimerase to ribulose. It means that in regeneration process, there are 4 xylulose 5 carbon cover. Xylulose 5 phosphate will be formed, and these xylulose get having epimers to ribulose 5. So here the xylulose which undergoes epimer into ribulose 5. So here 4 molecule of xylulose is there, means for example, if either 2 molecule means 2 molecule of xylulose converted into 2 molecule of ribulose 5 phosphate, means here xylulose converted into ribulose, that is epimer. So for that there is an enzyme is required. That enzyme is called epimerase enzyme. Clear? Right. So this statement is also right statement. Now coming to fructose combined with glycerolate 3 phosphate, transketolase and xylulose. Okay, so it means that here in this statement, here I'm generally out of 10 glycerolate 3 phosphate, there are first here 2 glycerolate 3 phosphate get converted or isomer into 2 molecule of DHAP, dihydroxyacetone phosphate. Okay, and one more 2 molecule of glycerolate 3 phosphate. What happens, see, and this DHAP 3 carbon compound, and here 2 molecule of glyceraldehyde 3 phosphate, both are combined together form 6 carbon compound, that is fructose 1,6 bisphosphate will form. And these fructose 1,6 bisphosphate which undergoes D process, it means it remove the 1 phosphate. So then it formed resulted structure is fructose 6 phosphate will be formed. So here giving fructose means fructose 6 phosphate, and these fructose 6 phosphate again combine with glyceraldehyde 3 phosphate in presence of this transketolase enzyme. Then it here 3 carbon compound glyceraldehyde 3 phosphate, 6 carbon compound fructose 6 phosphate, 3 plus 6 9. So here we need to, there is a formation of 9 carbon compound. Here 9 carbon compound will not there, so it is divided into to form 2 important component. One is 4 carbon compound, another one is 5 carbon compound. From this 9 carbon compound, here 4 carbon compound is nothing but erythrose. What? Erythrose. And 5 carbon compound here xylulose. So means xylulose is a product by here in this process. Okay, so this statement is also right statement. Okay, so right one is 1 2 3 and 4. So here next one, ninth question, consider the given equation. Here equation given, 2 molecule of water gives rise to 4 proton, oxygen and 4 electron. So where does this reaction take place? In the chloroplast. So this question which is framed from photosynthesis in higher plant, in that for mainly which is focused in photolysis. In light reaction there is a photolysis process will occur, so we will discuss here. Generally all these photolysis or photosynthesis process occur in chloroplast only. So here I need to draw chloroplast diagram. Chloroplast is a double membrane bound cell organelle. Okay, and in which we have sac flat like structure called like this thylakoid. What we have? Thylakoid. And each thylakoid here, each thylakoid which is made up of with 2 important structure. One is single thylakoid membrane is there, and inside the thylakoid membrane we have empty space, that is called lumen. That is lumen. Each thylakoid, if there is a group of thylakoid together form a structure called grana. Like that here also group of thylakoids together form grana, and these 2 granas are connected with a single thylakoid called stroma thylakoid. The empty space in the chloroplast is called stroma. Okay, fine. So these grana nothing but group of thylakoid, each thylakoid made up of with thylakoid membrane and lumen. See, this is what lumen. And remember, this thylakoid membrane is important, which contain thylakoid membrane contain a group of photosynthetic pigment, group of photosynthetic pigments, and these group of photosynthetic pigments together called photosystem. Okay, or simply we can use quantasome, or we can use quantasome. Photosystem or quantasomes are present on the membrane of thyla, and there are generally two types of photosystems are present: photosystem 1, photosystem 2. Photosystem 1 has maximum absorption P700 nanometer light, and PS2 has P680 nanometer, and that is mainly because of reaction center. Here also reaction means each photosystem again made up of with two important component. One is reaction center and LHC, or also we can use antenna. So here reaction center always made up of with a single pigment, that is chlorophyll A, but LHC, which is made up of with hundreds of pigments other than chlorophyll A. Okay, fine. All these are the important structure on thylakoid membrane. Now coming to lumen. Lumen, what happened we will discuss. So in lumen of thylakoid, very important process will occur, that is photolysis. So photolysis of water generally which is occur in lumen of the thylakoid. Very important. Lumen of thylakoid, or inner side of the thylakoid, or also we can use inner side of the thylakoid only. This photolysis process occur. Here photolysis also have various names, like it has water splitting complex, water splitting complex, or also we can use oxygen evolving complex. Why? Because here in which process oxygen is evolves, oxygen evolving process. We can use three name, either photolysis of water splitting complex or oxygen evolving complex. So what happened here in which photolysis? So for example, if there is a one water molecule and which undergoes photolysis, means then it produce two proton plus two electron plus half oxygen only. To produce one molecule of oxygen, there is a need of two molecule of water will undergo by photolysis. Then it produce four proton, four electron plus oxygen. Like oxygen is evolving during photolysis, hence this complex also called oxygen evolving complex. Okay. So finally, the right answer is, where does this photolysis process occur? Means in the lumen of thylakoid, not in the lumen of chloroplast. Okay, in the lumen of thylakoid only this photolysis process are occur. Okay, so next one, choose the incorrect pair. So now 10th question here, this one, choose the incorrect pair. Here in list given some various scientists and their discoveries in list 2. Okay, so Frankel, Conrad, Bessie, Buckner, Sumner, here their contribution. Okay, we will discuss few importance of this scientist, and but here we need to choose incorrect pair only. Okay, right. So first we have to identify which is right pair, and later we can eliminate by incorrect pair. Okay, done. So here about Frankel, Conrad. So he demonstrated that RNA is the genetic material of tobacco mosaic virus. So in tobacco mosaic virus, it is the first discovered plant virus, and this tobacco mosaic virus contain their genetic material is single stranded RNA. So who demonstrated the single stranded RNA which is present in tobacco mosaic virus? Means Frankel, Conrad. So this statement is correct. So and we need to know some important scientists with having contribution to this tobacco mosaic virus and tobacco mosaic disease. Just we will discuss what are the different various scientists they contribute to the tobacco mosaic virus. So here first we need to remember one important scientist called Dmitry Ivanovsky. Here Dmitry Ivanovsky. So he's the first person, he discovered disease, that is tobacco mosaic disease. He discovered tobacco mosaic, but he don't know so which is the causative agent for this tobacco mosaic disease. And his experiment was continued by another scientist. So he was considered as a father of virology. So he was Martinus Weisering. So Martinus Weisering continued Dmitry Ivanovsky experiment to find out the causative agent for this tobacco mosaic disease. Then he does many experiment. So finally he find out a fluid which is responsible for causative agent for this tobacco mosaic. That fluid name given as a Contagium vivum fluid. Contagium vivum fluidum. So he given the causative agent name as a Contagium vivum fluid. Means it is an infectious agent for tobacco mosaic disease, but he given the name as a Contagium vivum fluidum. And later, third scientist, most important, W.M. Stanley. So he renamed Martinez Beijering name, that is Contagium vivum fluidum, as a tobacco mosaic virus. So finally, who gave the term for tobacco mosaic virus means W.M. Stanley. But before tobacco mosaic virus, the name is Contagium vivum fluid, but it is given by the Martinez Beijering. Okay, tobacco mosaic virus name was renamed by W.M. Stanley. And not only that, but also he crystallized this virus. He crystallized this virus by using a method called X-ray crystallography method. He used who? W.M. Stanley, to know the structure of virus. So like that, to know the chemical structure of virus. To know the chemical structure of virus, this W.M. Stanley crystallized this tobacco mosaic virus by using X-ray crystallography method. And later, finally, this Frankel Conrad demonstrated and notified that RNA is the genetic material of tobacco mosaic virus. In RNA, generally we have double stranded RNA, single strand. But which RNA? That is single stranded RNA is the genetic material of tobacco mosaic virus, was found out by this Frankel Conrad. So this statement is right. Okay. Next, Bessey. So Bessey is he introduced phylogenetic classification. Okay, right. And this phylogenetic classification means to study evolutionary relations, evolutionary prospect. He proposed phylogenetic classification by Bessey. So this statement is also right. And coming to Buckner, important, Buckner discovered gymase enzyme. Here I am writing Buckner discovered gymase enzyme. It is the first discovered enzyme, very important. Buckner discovered gymase enzyme. It is the first discovered enzyme is gymase in from fungi. In which fungi? Yeast, very important. From yeast, here just wait. Yeah, so here from fungi. Which fungi? Yeast. So yeast is a fungi, saccharose unicellular fungi. Generally fungi are multicellular, but except yeast is a unicellular fungi, from which Buckner discovered enzyme called gymase enzyme. It is the first discovered enzyme. Now coming to one more important scientist here, giving about Sumner here. Yeah, about Sumner, James Sumner. And he discovered urease enzyme, very important. He discovered urease enzyme, not catalase enzyme. So this statement is wrong. So James Sumner discovered urease enzyme from Jock B. And do you know the James Sumner, he proposed, and he said enzymes are proteinous structures, proteinous nature. So enzymes are mostly proteinous nature. Statement was said by this James Sumner, and he discovered urease enzyme from the Jock B. So by observing all these scientists and their contribution, so C Sumner here, C D option is the wrong option. Why? Because Sumner discovered urease, not catalase. Okay, right. So one more. So 11th question here. The ratio of ATP utilization for carbon dioxide fixation between C3 and C4 plant. Here first we have to discuss. So there are some kind of C3 ATP utilization which is completely different C3 and C4. How means? Here to fix one molecule of carbon dioxide in C3 plant, it requires 3 ATPs. Remember, 3 ATPs. To fix one carbon dioxide in C4 plant, it required 5 ATPs. For example, to fix 6 molecule of carbon dioxide in C3 plant, it required here 6 into 3, so 18 ATPs are required. So to fix 6 molecule of carbon dioxide into C4 plant, then it required 6 into 5, 30 ATPs are required. Generally, to formation of glucose, there is a fixation of 6 carbon dioxide. For the formation of or synthesis of glucose, how many molecules of carbon dioxide fixation must and should be occur? 6 molecule of carbon dioxide. So in 6 molecule of carbon dioxide fixation, C3 plant required 18 ATPs, C4 plant required 30 ATP. But here in question asking only to fix one molecule of carbon dioxide, then it required 3 ATPs in C3 plant, 5 ATPs in C4 plant. So here in C3 now, then 3 and C4 required 5 ATPs. So here the right answer is second one.